Okay, so hello everyone, and welcome to today's webinar in which we will be discussing simulation for survival group sequential trials. My name is Brian Fox. I'm a research statistician here at nQuery, and I'll be your host for today's webinar. I will begin by just quickly going through the agenda for today. So I will begin with a brief introduction into group sequential designs. Then I will focus on survival group sequential designs in particular, discussing some of their complexities mainly. And I will set up an example of this type of trial, which we will then go on to explore and simulate from in the next section, which will be our survival group sequential design simulation section. I will be spending the vast majority of the time in this section and plan to go through some of the common areas where simulation will be used in tackling the uncertainty and issues that are common with these types of trials. I will aim to finish up with a brief discussion and conclusion, where I will recap on some of the main points from the webinar. Note that if you have any technical difficulties or anything during the course of the webinar, then do feel free to get in contact, and hopefully we can help resolve these. Also, just to mention that I will try to leave time at the end of the webinar to answer any questions that come through. But depending on time, I may not get to all of these questions, but I'll make sure to reply to any which I don't get to by email after the webinar. Also, if a question does occur to you after the webinar concludes, feel free to send us an email, and I'll give the relevant contact details for doing this at the end of the webinar. Also, just to mention that today's webinar is being recorded, and a link to it, along with the slides for this presentation, will be sent out to you shortly after this webinar concludes. Okay, so nQuery is obviously the sponsor for today's webinar, and it's the software that we'll be using to set up a lot of our examples later. So nQuery is widely regarded as being a cost-effective and easy-to-use trial design platform, which can be an essential tool in optimizing your clinical trial. It's known as the complete trial design platform to make clinical trials faster, less costly, and more successful. And nQuery has been on the market for well over 25 years. So nQuery is basically split into 4 tiers. Base being our kind of fixed term designs, Plus being our kind of Bayesian designs, Pro being our more adaptive trial designs, and then Expert having our prediction tool for both events and accrual. And mainly today we'll be looking at examples from both Base and Pro. Okay, so now let's finally get into the group sequential design part of today's webinar. Okay, so at a very basic level, sequential trial design is just one in which accumulating data is monitored and analyzed while the trial is still ongoing. You can either have fully sequential or group sequential designs. Fully sequential meaning that after each subject or a bit of information enters the trial, an analysis is conducted, whereas with the group sequential design, these interim analyses or looks at the data are conducted after groups of subjects or information has accrued. So at each of these interim analyses, the trial can stop early or adjustments can be made to the trial. The most common criteria for stopping will be stopping for efficacy and or futility. From a regulatory point of view, it's probably worth noting that the timing of these interim analyses and any efficacy or futility boundaries should be pre-specified in the protocol in advance. So the main benefit of catering for these interim analyses is that it provides a very large degree of flexibility to the trial. However, this flexibility does come at a cost, and we do need to account for the effects of conducting multiple interim analyses, as we are essentially giving our trial multiple chances to succeed, or well, also to fail, depending on the boundaries, which invites the issue of multiplicity of testing, by which I mean usual error rates will be naturally inflated, potentially. And this was shown in a paper by Armitage, in which they show that the type 1 error can be inflated from 0.05 to 0.142 from just taking 5 interim looks at the data. So multiple methods exist for combating this issue, and many of these take advantage of the fact that for each analysis, the data is essentially shared. So this provides an increase in efficiency compared to something like just using multiple comparison procedures. Okay, so what are these methods? Well, I won't spend too much time on these here, but effectively the one we're most interested in is the Landametz error spending approach, in which you are essentially taking your type 1 alpha or type 2 beta errors and spending a certain amount of them at each look, where the type 1 error is linked to stopping for efficacy and the type 2 error is linked to stopping for futility. The benefits of this is that it's extremely flexible and easy to use and adaptable. And so that's essentially why it's become the most common in practice. Other ones of note here are Habital-Peto and Wang-Ciatis. And also, just to mention, since it is a common issue, there are the classic designs of O'Brien, Fleming, and Pocock. But very often when someone refers to O'Brien, Fleming, or Pocock, they might be referring to the error spending equivalent of these functions. So it will be error spending a function equivalent of the O'Brien-Fleming method or the Pocock method. And indeed, later in the webinar, when I refer to O'Brien-Fleming, for example, I will usually be referring to the error spending approach equivalent. Okay, so now let's kind of move on a little bit, because I'm going to assume most people are familiar with all that, to more complex group sequential designs. And so the one that we're really interested in is the survival group sequential design. And so obviously, beyond just a fixed term design, or sorry, a fixed term survival design, having a survival group sequential design introduces even more complexity beyond just, say, a means or proportions endpoint, for example. And I will be discussing a lot of these complexities later, so I won't spend too much time on it here, but you can imagine that the patient follow-up, for example, obviously we don't know that in advance, and so it introduces an additional layer of uncertainty about when to conduct your interim analysis, for example. Okay. And then also, like, you can have other complex group sequential designs, like adaptive designs, like a MAMS group sequential design, which is shown on the bottom right here. But obviously, we don't need to worry about that today. And in fact, we do have other webinars on this topic. So if anyone wants to learn a bit more about those, just feel free to get in contact and we can share those, or they're also available on the website. Okay, so now let's get into the survival group sequential design part of today's webinar. So from our perspective, the most important thing to kind of discuss, since essentially nQuery is a trial design software and we're kind of worried about the power and the sample size, what we need to discuss is how to calculate power in a survival analysis or a time to event analysis. And so obviously the power will be affected by various different design choices. For example, whether a parallel design or a one-arm or multi-arm design is required. Also the statistical method or test chosen. For example, the log rank test, the Fleming-Harrington, the Cox model, etc. And in addition, the endpoint itself can have an impact on the power. For example, if you choose to examine the hazard ratio, or indeed progression-free survival or overall survival. And then finally, an important component of your power calculation will be the survival distribution chosen, where I suppose exponential is the most common usually, but viable or piecewise varieties of both viable and exponential are both very often used in practice. So one of the other kind of important things to note is that the power is not driven by the sample size directly, but rather by the number of events. And this is clearly shown in the formulae on the right here. For example, in the Lakine and Fuchs formula, which is commonly referenced in this area, one can see that lowercase e here stands for the events, and the formula does not depend on the sample size. So this also carries over to the group sequential setting, where the number of events will be linked to the information, the statistical information, and not being linked to the sample size. So our interim analysis as such will be linked to the events. Generally, you could also link it to the study times, but that obviously wouldn't be as advised, and we can look at that later within nQuery. Okay, so when it comes to sample size determination for these designs, one is essentially concerned with finding the sample size required to reach a target number of events while taking into account a certain time period. Given the potential complexities of these types of designs, sample size determination is often accomplished through a meta model, which accounts for the accrual process, dropout process, or crossover effects, etc. Or indeed, one can use simulation, which we'll see later in the webinar. Okay, so what are some of the complications that arise with survival group sequential designs? And which of these could be modeled using simulation to help alleviate some of their potential issues? Well, I'll spend quite a bit of time on this just to mention, since these will essentially form the basis of our examples later in the webinar. So an obvious complication is that since we don't know exactly when each subject in the study is going to have an event, or the follow-up time is unknown, it may be difficult to predict when an interim analysis will occur. For example, if you take a look at the top right image here, which is from nQuery, you can see the issues that would occur if the first interim analysis was expected to be around time 2.05 roughly, after 45 events have been observed. But at time 2.05, say we only had 20 events, due to the fact that the dropout rate might have been significantly higher than what was expected, for example. And then this analysis might be significantly delayed, and potentially there could be a very real chance of the trial being underpowered and failing to find efficacy, even if there was a true underlying effect. And indeed, even from like a logistical perspective, this could be a nightmare, as you can imagine. So even just beyond the uncertainty of when these interim analyses will occur, you also have the additional complication or decision of whether to examine progression-free survival or overall survival, as is illustrated on the bottom right here. Okay, so additionally, higher numbers of events are quite likely in the active cohort, so at each of the interim analyses, so you'd have to likely take that into account in your analysis. Also, additional complications can arise if you're planning to conduct an unblinded sample size re-estimation, beyond just the usual kind of practical considerations associated with unblinded sample size re-estimation itself. For the time to event endpoint, there are additional kind of trial integrity considerations to be made. For example, Friedland and Korn in their 2017 paper discuss how access to interim information on the shape of the survival curves may actually jeopardize the perceived integrity of a trial if you're using a sample size adjustment design. Okay, so it's also worth mentioning that survival group sequential designs may require a lot of additional assumptions. And so modeling and verifying these assumptions is where kind of simulation can become so invaluable. So what are these assumptions? Well, an example would be, should there be a constant treatment effect assumed, or non-proportional hazards present? And indeed, a common issue in survival analysis is the issue of stratified analysis. For example, a fixed sample size design can simply use the Mantel-Hansel log rank test or something similar, or indeed actually the Cox proportional hazards regression model. And I should say that, it has been shown that the sequence of tests from that model can be applied to an independent and normally distributed increment structure, which in turn shows that it can be applied to the theory of group sequential designs, although it does obviously complicate matters somewhat. A final point to note here is that there are two main avenues that one can use to try to increase the number of events at each given interim analysis. One may increase the sample size in order to increase the amount of subjects available to have the event. However, this can introduce a bias towards earlier event trends, or one can increase the time taken for an interim analysis to take place, which can introduce a bias towards later trends. So it's quite important to keep that in mind. So how does one combat all these issues? Well, this is essentially where simulation can be extremely useful. And indeed, almost every one of the issues mentioned here can be alleviated somewhat, at least, through the use of simulations. Okay, so finally we can get into an example. So this particular example is for everolimus for advanced pancreatic neuroendocrine tumors. And this is an example which we've looked at many times before in our webinars here at nQuery. So hopefully some of you may be familiar with it. Essentially, like the majority of this isn't really of interest to us. All we really need to worry about is the fact that that we expect we need, based on the study protocol, a total of 352 patients, allowing for or expected to receive 282 progression-free survival events in order to gain 92.6% power. So we can now switch over to nQuery, and we can see that here we have essentially replicated that example. I've done all this beforehand to save time, since what we're most interested in today is survival group sequential design simulation, but it's important to go through these just to set up the example. So you can see that within nQuery, for those of you who aren't aware, there's essentially three main sections to our design tables, we like to call them. We have our main table up here, which has kind of our main parameters that are of interest. So you can imagine the power, sample size, the significance level, these will all be very common. And then we have a side table to the bottom here. In this case, it's kind of additional survival parameters that you can enter to get back your group 1 and group 2 exponential parameters. And then on the right here, we have help cards, which will give you a brief kind of summation of each of these parameters. Apologies, my laptop is a bit old. But in order to open a lot of these tables, as we like to call them, we have this table select tool, which has actually been updated in our most recent update to the software. So we will have webinars discussing this, but just to quickly touch on it, we now have a slightly different way of accessing our tables. So you can choose to search. So you can search for log rank, not Lego rank, log rank. And you can see that will bring up relevant tables, or you could go and click on survival, etc. Just to quickly show that. It's quite useful. It also is split by adaptive design and fixed term designs and Bayesian designs. So if you wanted group sequential designs, for example, you can click here and all our group sequential tables become available. So it's probably just worth mentioning that. So in our case, we're looking at this table here. And as mentioned, we have our 92.6% power. We have our sample size per group of 176, and we have our total events of 282, which, if we go back, matches what we expected. So just to quickly mention it, within nQuery, rather than just simulating a group sequential trial, you could also use this table here, the SDT tree, which allows you to simulate for a two-sample log rank test. And you can specify your own piecewise rates. For our example, we're actually looking at this one here, And we can see that due to the fact that simulation is a little bit different, but we're still getting a power of 92.6% for our sample size per group of 176. Now, just to quickly mention it, there's also this table here, which is a little bit more complex and allows for fixed follow-up, allows you to change the variance hypothesis from the null to alternative. You can have rounded sample size error events, and you can have piecewise dropout, etc. So just to mention that that's also available. Okay, so now let's go back and let's extend this original survival example, or fixed term example, to the group sequential setting. So this is where things get a little bit more complicated, a little bit more interesting as well. And this will be quite important later when we come to simulate this example. So I'll spend a bit of time going through this. So essentially, we're going to be hypothetically extending the original example to what might have happened if they had chosen to do a group sequential example. So, oh, my apologies. So here on the left, we have our additional assumptions that we're making in order to extend this to a group sequential example. So we're going to be assuming that there are 4 equally spaced looks. So by equally spaced here, I'm referring to the information fraction, and that will become a lot clearer within nQuery in just a moment. We're going to have both efficacy and futility boundaries, where we're going to be using the Landemetz, oh, my apologies. We're going to be using the Landemetz error spending approach, that's the LDM here, and we're going to be using a non-binding futility boundary. So the alpha spending function we'll be using is just the O'Brien-Fleming analog. So once again, I think I mentioned this earlier, but it's going to be the O'Brien-Fleming analog within the Landemetz error spending approach framework. And then similarly for the futility boundary. I'll show this one in nQuery later, but there are multiple other spending functions that you could choose. But yeah, it's not the main topic for today's webinar, so I won't spend too much time on that. So the survival time periods that we'll be using are essentially 6 months, 18.5 months, and 28.25 months. And these are effectively what you get if you change the accrual period, minimum follow-up period from weeks to months. But yeah, once again, we don't need to worry too much about that. We're more focused on the group sequential issues. So for our placebo exponential hazard rate, we're going to be assuming that's the same as before, so 0.077, which is taken from nQuery, if you remember. It's pretty easier to show it here. It's this value here. And we're going to be assuming that to be initially at least the same at each of our piecewise survival time periods. So we'll be assuming that it's the same from time 0 to 6, 6 to 18.5, and 18.5 to 28.25. And I'll explain that a little bit more later. We're going to be assuming a dropout rate in the control group of 0.03 from time 0 to 6. And then we're going to be assuming that it's 0 after that time in the control group. And then for everolimus, we're going to be assuming that there's no dropout until time 6. And then between time 6 and 18.5, it's an exponential dropout rate of 0.03, and then it's 0.01 thereafter. Okay, so I know I spent quite a bit of time on that, but hopefully that explains the assumptions that we're going to be making. And now we can go into nQuery, and we will see that we have this table here. So this is our group sequential table, or our new ish group sequential table for survival curves, or 2 survival curves. And this is a very flexible table, which essentially extends this table here, STT37, into the group sequential setting. So we have multiple webinars already on how to use our new group sequential tables, so I won't spend too much time on that here. But essentially, we just have our group sequential parameters down here, or well, the majority of them. We also have to specify our spending function, or sorry, our efficacy boundary calculation method and utility boundary calculation method, which we're going to be using the spending function approach as mentioned. So that's the Land-Metz error spending approach. And we're going to be assuming 4 looks here. We have 3 periods, if you remember, which is in this tab here, this piecewise survival information tab. And so we have 6, 18.5, and 28.25. And you can see that we've assumed our dropout rates specified down here. So now we can essentially, so I'm not going to run it now because it takes a tiny bit of time, but now we can see that effectively there's been an increase in sample size from 176 per group to around 216 per group. With the fact, sorry, this, I should mention that this red numbering here is just an indication that the total study time has been increased. And I can show that here. So it's an indication that the total study time has been increased under the null hypothesis, as you can see here, from the initially specified 28.25, it had to be increased up to 32.69 in the under the null hypothesis case in order to get our total number of events. And so a warning is just output in nQuery whenever that happens. But yeah, that's nothing really to worry about here. So, okay, so yeah, hopefully that gives you some idea of what a group sequential table is like within nQuery. As mentioned, we have lots of webinars on it. But if you do want to learn more, just let me know and I can send those on. Okay, so now let's go back and get into the simulation part of today's webinar. So yeah, so just to quickly define it. So by simulation here, I'm referring to the repeated execution of a large number of hypothetical clinical trials to understand operating characteristics of a trial design under a series of specific configurations of assumptions. So that's very exciting stuff, I know, but it's hopefully useful just to make sure that everyone's aware of what I'm referring to by simulation, and to note that it's distinct compared to something like modeling, or something like, Monte Carlo or bootstrapping methods, where you might be trying to extend a data set, for example. So yeah, hopefully that helps. So simulation is increasingly used, although not traditionally thought of, as part of the clinical trial design process. And it is not intended to replace, but rather to kind of supplement a closed form statistical analysis. And from a group sequential perspective, the simulated study data can be extremely useful in predicting a lot of group sequential design characteristics. And obviously, this is particularly of note with more complex group sequential designs, where one might want to verify the kind of closed form design that's being considered, by which I mean you might want to verify the type 1 or type 2 errors, the expected sample size at each interim analysis, or overall. You might want to verify the expected time at which the group sequential trial should stop, and indeed, under both hypotheses even. And indeed, it can be thought of or well used directly in the group sequential boundary calculations, although that's, I suppose, a bit less common. So another very useful aspect of group sequential design simulation is that it allows you to very quickly explore and evaluate other designs or study scenarios. For example, like what would happen if the treatment effect or hazard ratio turned out to be far larger or lower than expected. And how would it affect the interim analysis timings, or the likelihood of stopping early, or the expected sample size, etc. So it is also very useful, or simulation is very useful, I should say, to explore different nuisance parameter values or assumptions, like the standard deviation, for example. Obviously not of note in the survival design directly, but this is very powerful and a very useful aspect of the of these types of analyses. So it's also worth mentioning actually that if you've made any like large sample assumptions, simulations can be very helpful to verify these. They can also help facilitate comparisons between adaptive and non-adaptive designs and help you decide if a group sequential design is actually even necessary, for example. So simulations also allow you to explore what would happen if the interim analyses or information fractions turned out to be different to those in the original design. Excuse me. So this is extremely useful, as you can imagine, since trying to predict when these interim analyses will actually occur with any degree of certainty can be very, very difficult. And due to the nature of group sequential designs, changes to the information fractions can have very large impacts on the resulting boundaries and error rates. So this is a major benefit of a simulation for these types of designs. Finally, it's worth noting that simulation avoids a lot of the constraints associated with closed form analyses, where, for example, it might be very difficult to model a complex crossover like multi-arm design or something like that, or indeed a closed form method might not even exist, whereas a simulation method could easily be developed, or well, relatively easily. Okay, so I suppose I should also mention that due to their nature, simulation studies are very easy to explain or convey to non-statisticians and can be very useful in discussing with other stakeholders. For example, it's quite easy for most people to understand that out of 1,000 simulated studies, 100 stopped for efficacy at the first interim analysis, and so that the probability of stopping for efficacy at the first look is 10%. Also, actually, I suppose I should mention that simulations can inform, internal sponsor decision making about actual trial logistics, such as like what site to select and what drug supply to use, etc. But yeah, I suppose that's probably enough time spent on that. So let's take a look at this from a regulatory point of view. And I won't spend too much time in this, but I suppose the news is that luckily, by and large, the regulatory perspective on simulation is very positive. The FDA themselves view modeling and simulation as an important part of the regulatory review process, and this view is shared by many other regulatory agencies. So the multiple guidance papers on this, I should mention, I've included some of those in the references below. And also the EMA has hosted multiple workshops on modeling and simulation, and the results of some of these are included in the references. I suppose the main thing to mention from a regulatory perspective is that regulators tend to just stress the use of standardized modeling and simulation methods, accounting for issues such as internal or external validity, and the control of bias and the type 1 error, especially in a confirmatory setting, obviously. And I suppose an area where simulated studies can be of particular benefit, as mentioned earlier, is that of adaptive designs. And indeed, actually, the recent draft guidance contained in ICH E20 clearly summarizes and outlines the benefits of simulated studies for adaptive designs. And I included some of the just key points that was raised in that guidance here. But yeah, I won't go into any detail on that, but it's included in the references, and it's very useful for anyone who's involved in adaptive trials, so I'd really recommend it. Okay, so, so what are the steps in actually designing a survival group sequential design simulation? So these can essentially split into 5 steps. The first will be your generating model assumptions, so stuff like your sample size, your accrual rates and processes, etc. The next would be your survival design assumptions. So these would be things like your type of follow-up, do you have fixed follow-up or maximum follow-up, etc. How many time pieces, or sorry, how many piecewise accrual or dropout or event processes are there, and your event process, is your hazard ratio constant, or are there non-proportional hazards, etc. And also, similar for dropout rates. And I've shown a few excerpts from nQuery here, or screenshots from nQuery, I should say, in case they're of use, but obviously we'll be looking at these in just a moment. So I suppose there are certain questions that you would have to keep in mind when you're designing a survival group sequential trial simulation. And these include just how to justify your population assumptions, like how to justify the accrual process, how to avoid extrapolation issues. And this is usually done through the use of a standardized modeling and simulation method, as discussed in ICH 820 and other regulatory documents. And finally, how to model the survival or accrual process, which is a pretty obvious question, but one worth noting, I suppose. Okay, so I've probably spent plenty of time on that, and we can finally get into a survival group sequential design simulation example. So this is just a replication of the example that we were looking at earlier, or the group sequential example, I should say. Since from a simulation perspective, this is kind of all we need to worry about for the moment. I'll kind of discuss the assumptions I'm making, which are related to the simulation, as we go through it. Okay, so now let's have a look at this within nQuery. So within nQuery, you can access our group sequential design simulation tool essentially in two ways. So for our modern group sequential design tables, we have this button here. So if I click this, we can see that our simulation tool opens, and any parameter values which were present in our original example, so you can see our sample sizes of 216, will be carried over into our simulation example. So if I was to continue through here all the way, and I'll be going back to discuss these, don't worry, but if I was to continue through here all the way and run the example, we should get back essentially a very similar example to our fixed term design. But here, we're going to, I'll spend some time discussing each of these steps in a little bit more detail. But before I do that, I'll just mention that you can also access this tool through the assistance menu here. And if you use this, obviously, we have our other endpoints which are available. And if you click on survival, you see that it just opens up a blank simulation tool. But yeah, we don't need to worry about that today, because it takes a bit of time, obviously, to type in all of that. So if I go through here and kind of explain just a little bit of this that might be of use, you can see that originally we had time periods of 0 to 6, 6 to 18.5, and 18.5 to 28.25, whereas here we just have time 0 to 6 and then nothing after. So the reason for that is that within the group sequential design, we've assumed that our accrual finishes at time 18.5. So with our simulated study, we're not going to know the end time. So we don't know exactly when the trial is going to finish, because these are all simulated trials. We can't say exactly. So instead, what nQuery does is it will assume an accrual rate based on the implied accrual percentages here. So that works out as an accrual rate of 1.8 from time 0 to 6. So 6 by 1.8 should give us our rough accrual percentage, so 2.5% by this. And then after that, our accrual rate of 33.7 roughly from time 6 means that we expect the accrual to finish roughly around time 18.5. So yeah, that's essentially all that is. So on the left here, the first few of these parameters are very simple, so I won't discuss those. And by randomization here, this is also very simple, but this is just the type of subject randomization that's going to be used within the simulated study. So fixed by ratio assumes that the sample size ratio is fixed, whereas complete randomization will randomly allocate subjects to the control or treatment group based on that sample size ratio. So it's not fixed. Okay, so now let's move on to step 2, where we can see our piecewise event and dropout tables here. So these are quite self-explanatory. These are exponential rates, I should say. And we can see that our hazard ratio is constant at 1.5 all the way through. And these are the same rates as we found earlier, where once again, we're saying that this hazard rate of 0.077 for group 1 applies from time 18.5 until the end of the study. And similarly for the dropout rates. Okay, so you can also choose for the follow-up type to be event-driven or to have fixed follow-up. So event-driven essentially meaning that every subject is followed until they have an event, or dropped out, I should say. Whereas if you choose fixed follow-up, you can choose your fixed follow-up time there. But we don't need to worry about that here. Okay, so now if we continue, we get to our kind of group sequential design inputs. And the first choice to be made, is actually more related to survival, but affects the group sequential design, obviously. And that's to choose your test statistic that will be used to compare against your boundaries. So the first choice is the log rank, which is obviously the most common. We also have the Wilcoxon test statistic, I should say, and also the Fleming-Harrington. Obviously, if you choose Fleming-Harrington, you can choose your parameters to define that down here. And we have good webinars explaining how to use Fleming-Harrington method if any of you are interested in that. But we'll just be using log ranker today. We have a one-sided test. We have 4 different looks, so that's 3 interim looks. And we have our efficacy boundaries being on the Z scale. You can also choose the p-value scale, and then the futility boundaries similarly will be assuming them to be on the Z scale. And then, as I mentioned earlier, you can actually change your interim analysis criteria, by which I mean the criteria for deciding when to conduct an interim analysis. So the events is more closely tied to the idea of information, which is essentially the bedrock upon which group sequential methods tend to be discussed. But you can also use analysis time, and if you choose that, you can just say that your interim analysis will occur after, a time period of 2 months, for example. But in our example, we're going to leave that at events. And when you go in, you see that this results in interim events of 76, 152, and 228, which is essentially information equally spaced information times or information fractions of 0.255 and 0.75. And then here we also have our efficacy and futility boundaries. Once again, I'm going to hope or assume that many of you are aware, are familiar with group sequential boundaries, so I won't discuss those. But yeah, do let me know if you want me to send on any information about them. So now we get to our simulation control step, in which we can define how many simulations we want to run. So I'm just going to run a very small amount, only 1,000, and we're not going to save any data sets. So essentially the summary statistics will give you a summary of each simulated study, a summary of all the simulated studies for every run. And then the subject level data will give you every single bit of data that's being put into the simulation, essentially. So yeah, and we could specify a random seed, but we don't have to. This will be chosen at random if you don't. So if I click run, we can see that this box pops out. And essentially, this is our just calculation in progress window. And while the simulation is running, we see that we get updates on the current average sample size, events, dropouts, study length, and then also our percentages which have stopped for efficacy and our percentage that has stopped for futility. So once this finishes, which should be pretty soon, hopefully, we can see that we get this, which is called our simulation report. So obviously we only ran 1,000 simulations, so this isn't going to be the most robust simulated estimate, but we can still see that we get a power of 91.4 compared to our expected 92.6. But just for the sake of time, I actually have one of these already ran, just here, which is with 10,000 simulations. So you can see that here. And so for the purposes of comparing against our original group sequential example, I'll be using this. So here we can see that we have a power very close to 92.6. So this is the average power, which is essentially the number stopping for efficacy, since that is the power under the alternative hypothesis situation, which is what we're simulating here. We can see that our expected study time is roughly 28.25. So very, very close. You can see that we get the total number of events. And so just to compare this to the original group sequential design, we have study times of 13.5, 17.5, 21.33. And if we go in here and look at our group sequential report, you can see that hopefully, yes, matches on pretty closely to what we expected, which was, so this is the alternative hypothesis information table, I should say, to 13.6, 17.5, 21.48, and 28.25. So you can see they're quite close, or quite similar. And then also we can see our per group events and dropouts, which we expect to be 30 and 48, 62, 89. Obviously, I'm not going to go through every value in the table to let you know. But if you add them up, you'll see that these very closely map on to the expected events and dropouts here under the alternative hypothesis. So yeah, we can also check our expected sample size and events under H1. So under H1, we're expecting 218. H0, we can't look at that yet because we haven't simulated it. And then our expected sample size is expected to be 432 in both cases. In the group sequential setting, it will be a little bit lower, I'd expect. So we're getting 420 because we are stopping for efficacy and futility quite a bit here. But yeah, hopefully that kind of explains it a bit. And so yeah, other things worth mentioning here. Obviously the boundary table, that's all just our inputs. We don't need to worry about that too much. That's just related to the group sequential setup. And then a lot of the outputs on the left here are just summaries of what's being included, except for these allocation parameters, where we can see the average total events was 220, which is very similar to our expected events, which was 218 roughly. And also the average dropouts and accrual period length, which is 18.15, and we expected it to be 18.25, sorry, 18.5 based on this specification here. So we can see that the accrual process is working correctly, which is very good to know, as you can imagine. And so, yeah, there's multiple other things you could be looking at. Obviously, your average sample sizes are very important to keep track of. And then, yes, these are just summaries of the kind of group sequential inputs for the accrual, hazard, and dropout processes. So, yeah, I hope that was of use now. That's just like a basic summary of what the group sequential survival simulation table outputs in its report. Obviously, you could spend a lot of time going through all this, and I'm not going to do that here. You might get a bit bored. But that's just a highlight of some of the really useful aspects of it, which we of use from a regulatory perspective as well. But also in nQuery, you'll also get a rejection plot, which will show you the percentages, or sorry, the counts of how many of the trials stopped for efficacy or futility at each look, or indeed how many continued until the next look. So you can see here that very, very few have stopped for efficacy or futility at the first look, due to the fact our boundaries were a bit pessimistic, despite the fact they were O'Brien and Fleming. They were being a little bit pessimistic. And then as we move forward, we see at the final look, obviously a decision has to be made. And so you can't continue beyond this look, and you have to stop for efficacy or futility. So yeah, that's the rejection plot. We also have our boundary plot, in which you can see we have our efficacy bound region up here in blue, where if the interim Z statistic or test statistic, I should say, at each interim look is above this point, then a simulated trial would stop for efficacy. Below, in this red region, we'd stop for futility. Pretty basic stuff. And then also, we have our summary data per simulation. So this will effectively give you a summary of each simulated trial. So you can see that the first simulated trial here, given simulation 1, stopped at look 2. And if we move across, we should see that its test statistic was 4.1, which is obviously above our first boundary. So we can see that we stopped for efficacy. But yeah, hopefully that is of use. What we can do here is we can explore the question, excuse me, we can explore the question of the type 1 error rate. So say we wanted to think about what would happen if the null hypothesis turned out to be true, then we can go in here, we can press this again, and now at step 2, instead of having our hazard ratio be 1.5, we can set them all equal to 1. Oh, apologies. So if we were to run through with this until the very, very end, obviously I'm not going to run it because it does take a bit of time, but I do have it around here, I believe. I'll just check. Yeah. So you can see our hazard ratio is 1 at each look, and everything else has remained the exact same. And you can see that we get back our power of 0.23, which is very similar to what we'd expect it to be, which is the type 1 error rate of 0.25, since this is the case where the null hypothesis is true. And so we can see that the total number stopping for efficacy is 230. Obviously, in this case, your boundaries essentially, this isn't kind of meaningful use of the boundaries for planning a trial, but it's extremely useful for verifying that your error constraints are being met. And you could run this with far higher degree of simulations, so far higher than 10,000 if you wanted to be even more sure. Okay, so now let's take a look at the issue of non-proportional hazards, let's say. Or, well, what would we do if that that assumption didn't hold? And so we can do this by, once again, going back, creating a new simulation. And at the same step, so step 2, we could say that the hazard ratio just happened to be a lot higher at at the second piecewise process. So if we were to change it to 1.8, and once again, if we continue all the way through, we would get, I believe, this here. And what we find, if we assume that the hazard ratio is even higher than expected at step 2, we can see that we get a very large increase in the power. And so you can see that the case of the non-proportional hazards not holding, well, to the degree of 1.8 for the second hazard ratio, can have quite a significant impact on the trial. Obviously, in this case, it will be more beneficial since it's a higher hazard rate or hazard ratio, which we're assuming to be positive. But this can have a very significant impact, and so simulation can show the effects of that non-proportional hazards very, very quickly, and could help you verify if you're using like at the simulation the test statistic step. If you were expecting there to be non-proportional hazards or any kind of crossover effect, then you would probably end up using the Fleming-Harrington test statistic. And simulation would help you to verify that using that test statistic is more beneficial to the design. So it's extremely useful for that, where obviously complex survival curves or anything like that can be quite difficult to manage with a closed form solution. And indeed, since I've mentioned that, we can think about what would happen if, once again, if we went through and we changed these hazard ratios to having a flipping, to there being a flipping of the hazard ratio. So for it to cross over 1. So if we did that, we can go into this example here, which I ran, where the hazard ratio has changed from 1.5 to 1 to 0.5. And you can see that in this case, there's a very, very large reduction in the power, which obviously you'd expect. In fact, when I was running this, I would have thought it was even larger reduction, but you can see it's enough that it's a massive difference. And the simulated sample sizes and study times are quite heavily affected. So you can see what's expected to happen if this turned out to be the case. If the treatment effect was tailing off in the treatment group towards the end of the study, let's say. So yeah, just conscious of time. So one last thing that's probably worth mentioning. Well, actually, before I do, obviously in this case, you would probably have specified Fleming-Harrington 1-1. Fleming-Harrington 1-1, I believe, test statistic. But anyway, yeah, just worth mentioning that. Okay, so now let's say, rather than just being very preoccupied with the hazard ratios, as I was, that we want to test the accrual process. So in this case, we can once again go back, use our simulation button, and we can think about what might happen if, let's say, there was a very high level of toxicity, which we weren't aware of in the treatment group, or that the phase 2 trial hadn't, or might have indicated that that was going to be the case, especially when the drug is being used for a long time, let's say. So what we would expect is that there will be a higher dropout rate in group 2, which is our treatment group, especially as the study goes on. So let's assume that instead of these, we're going to have 0.05, 0.095, and 0.11. So if we were to continue through with this, what we should get, or sorry, what we would get, is something very similar to this here, where you can see I've already ran this example, and you can see that we get actually only a slight reduction in power, but that there are far higher numbers of subjects dropping out. It does affect our study times. You can see a study time of 43 here, which is very highly inflated. And indeed, the sample size are also affected, but to a lesser degree. And you can see here that our percentage of trials reaching the target events, so reaching our events of 304, actually isn't 100%, which it was in every other case. So you can see that we have 304 here, or sorry, 303.97. Obviously, 304 is what we're aiming for. So if you were to go through this simulated data set, I'm not going to have time to do it, but effectively, you'll find that some of these total sample sizes at the final look won't add up to what you'd expect. Oh, my apologies. Or well, actually, we're basically done with nQuery now, but yeah, hopefully that gives you some idea of the power of simulation, how easy it is to use. You can see the majority of these parameters are quite simple and kind of easy to understand, and it's more kind of decisions about how to set up your simulation. So now let's briefly conclude with a brief discussion and conclusion. So once again, just to mention, group sequential trials, we're all aware of what they are, but they just facilitate interim analyses and interim decision making. And in real life, there can be many different complexities added to the group sequential setting, in particular in the survival case. And so in the survival group sequential design case, one might require a lot of different considerations or assumptions, and some of those are shown here. And verifying these assumptions, or trying to predict the issues that might arise from them, both for a trial itself and also from a regulatory point of view, can be very difficult. And this is where simulation can become very useful, because it allows verification of not just the operating characteristics, but also of alternative scenarios, around the dropout rate, as we just saw. And it's very easy to understand and convey to non-statisticians and also to iterate upon. So yeah, extremely useful, extremely powerful for something that's quite simple at its heart. And then also just finally mention, we know that regulatory guidance on simulation for survival group sequential designs or adaptive designs in general is actually quite positive. Okay, so quickly before we finish up, I'll just mention that you can go to statsalz.com/nQuery demo if you do want to get a demo of any of the things you've seen here today, or do feel free to get in contact at info@statsalz.com or just visit our website if you'd like for more information. So thank you very much for staying with me all this time. Hopefully we're just about out of time, so I won't get to any questions, but do feel free to send them on and I'll get to any that have come through there shortly after this webinar. So thank you once again, and bye.